So I will tell you a little bit, about what are the stem cells, in which circumstance you can find stem cells, what kind of disease can be associated with stem cells, and what kind of the disease you can treat potentially with stem cells, and a little bit what is the future, or what I can see to be the future of stem cell biology. So the stem cell is the mother of all cells. So it's sitting on the hierarchy, on the top of the hierarchy of all cells within a tissue. And it had two properties. The first one is to renew, so make another stem cells. And the other properties is to differentiate. And differentiate, it means to adopt the morphology of the different and the molecular feature of all the cells that compose the tissue. That's called differentiation. And sometime in between the stem cell and the differentiated cells, there's what we call transient amplifying cells, so the cells that expand the pool of cells to make the different cells that compose our tissue. And the stem cells come into different flavor. So the most primitive stem cell is called pluripotent stem cells. So that's called embryonic stem cell as well. And these cells can give rise to all cells of the organism. I will show you their role and what we can do for that. There's embryonic progenitors, so during development, the different tissues are formed, and that's also stem cells that are at the basis of the formation of the organ and the tissue. And then, during our adult life, most of our tissue constantly renew throughout the life. Our gut renew in 5 days, our hair is, or the skin is renewing 2-3 weeks. Some blood cells renew, almost every 2 or 3 days. And so, to renew all these cells that die constantly, you need cells that divide and replenish the pool of cells that disappear. And that's the adult stem cells. And another part I'm going to talk to you about the cancer stem cells. So we use somehow the knowledge that has been gathered in all these normal stem cells and try to see whether cancer somehow is a little bit like a perversion of the thing that normally occur. And all these data, if you are more interested to go into the more detail, I'm giving a cycle of conference at the VUB. It's called Share Frankie this year. And so that's the different things. But today you're going to have a condensed view of all this lesson in one single lesson, so that's even better. So the pluripotent stem cell, the embryonic stem cell, what they are, those cells? Those cells are, that's a egg that's been fertilized, and after the spermatozoid encounter the eggs, there's the cell divide in 2, in 4, in 6, in 8, and until they reach, about several hundred of cells. And at this stage, it's before the implantation of the embryo, when the implantation is not yet in the uterus. And at this stage, in the yellow part here, it's what is called the epiblast. And the epiblast, you can take those cells and put them in a petri dish and capture them in their very early states, in which everything is possible, because these yellow cells here will give rise to all the cells of the animals here, all and every single one, but the placenta. That's the only cell that they don't give rise. And so you can derive those cells, culture them in the petri dish for years and years and years, and then reimplant them eventually into the embryo, and they will resume development and incorporate in the embryo. Of course, you cannot do that with the human cells. You do that, with the animal cells. And that has been, very, very precious and important to study many human disease, many mechanisms of the different genes that compose of our body. And I'm going to talk to you about, how it's possible when you differentiate into you take adult cells and you can revert them to their most primitive cell type. And this concept, which looks completely crazy, is to revert back the time from adult to embryo, the first day where you have been created. That has been awarded by the Nobel Prize in 2012 to these two gentlemen, John Gurdon and Shinya Yamanaka. And John Gurdon actually begins the proof of principle that you can revert back and clone animals. And so how he did that, it was in the 1960s, very, heroic work, where he could take from a frog a cell, a nucleus of the cell. So the nucleus, as you know, contains all the genetic information of the animals. And take eggs that has been fertilized, remove the nucleus, and put back in these fertilized eggs a nucleus of a very adult cell. And this nucleus is being reprogrammed into the most primitive cells, and it resumes completely the development. And so all this frog that you see here is a clone of these frogs that has been cloned, where the nucleus has been cloned. And so somehow that, this image where you revert back the time and you go back to the pluripotent cells and they resume the development and differentiate. After the demonstration of this possibility that you can clone animals, Jan Wilmut was the first scientist at the University of Edinburgh in Scotland to clone mammals species, and he cloned this sheep that's called Dolly. You all know, Dolly, and Dolly, and give to kids, and since Dolly, basically almost all mammals that you can think about have been cloned so far. Cow, dog, pig, chimps, cats, and even they clone at one point a mammoth and this ancient elephant, and put them back into an elephant mother, and it almost gives birth to and unfortunately just a few weeks before giving birth, the baby died in utero and could not resume. But otherwise, the Jurassic Park kind of thinking has been real, it's been there. And what Shinya Yamanaka did, it was a different kind of approach. He said, well, what are the genes, what are the information that are in these pluripotent cells? And if I, put this gene back, this embryonic gene back into a very differentiated cell type, can I reset the clock and go back to this embryonic early primitive state? And it really works, and it was really one of the most, groundbreaking discovery of the last decade, is to demonstrate that with 4 genes only, you can reset the clock of your most differentiated cell type in your body and send back. And so one of my, and no, it's very easy to do that. Every one of you can do that within, 2-3 weeks. It takes that, and I've, some friend of mine who are, professor of stem cell biology in different university, who ask their student to clone, to make their own embryonic stem cell from their own, own cell type. And so of course, why is this, so important to have this possibility to reprogram to go back in time and make your cells revert back to their pluripotent cells? Because these pluripotent cells can give rise basically to all the cell type of the body. So of course, if you take a patient, you reprogram them and you make iPS. iPS is the name to induce pluripotent stem cells. You can, mimic disease if this patient has some disease, and you can model somehow disease in vitro, and then try to define why these cells are sick, and can we find new drugs to help those patients, and so that's it's for disease modeling and drug discovery. But also you can use this potentially, and I will show you one example that's that's already in clinics, is to use these cells and differentiate it to, let's say, beta cells if you suffer from diabetes, from cardiac cells if had a big cardiac disease, or some neuronal cells if you, for example, suffer from Parkinson's disease, and graft them back to the same patient. And so somehow you don't then have to deal with immunosuppression, you don't have to deal with rejection. It's it's your own cells that you retransplant. You just redirect them and re-engineer them into the particular cell type. And so in my lab, we are interested in understanding how we can make from pluripotent cells one of the most important cell type in our body, which is the cardiac cells. You know, the cardiac organ is the first organ that developed during embryonic development, because as soon as the embryo grows, you need to give nutriment and oxygen to distance cell type. And so for that you need a flux that brings them all the goodies that they need, the oxygen and the nutriment. And the cardiac cells, as you know, is very important. Another why it's so important is because cardiovascular disease are probably the leading cause of mortality and morbidity in our Western country. And so, very frequently patients, develop after myocardial infarction or other type of disease can suffer from cardiac insufficiency. And so, understanding the mechanism to recoax the cells into cardiac cells is very important. And so we discovered, back in 2008, really the molecular switch in which when you put this gene, a single gene, into an embryonic stem cell, this pluripotent stem cell that I just showed you, all cells differentiate into cardiac cells. And so I will show you a little movie. So Antoine Bondu, who is now the head of the cardiology at the Hôpital Erasme in Brussels, when he was a PhD student in my lab, he developed this technique in which we can turn on this gene. And what he found, it was spectacular. When he turned this gene in pluripotent cells, you see here these beating cells in the dish. I will replay the thing because I cannot. You see these little beating cells. It's all cardiac cells that beat spontaneously. So when he looked at this microscope and after turning the gene, he knew that he had hit something important. And so after that, we dissect out the molecular mechanism to why these cells were, beating and were differentiating into cardiac cells. After that, what Antoine did is try to define the way to isolate this early cardiac progenitor, the one that will become cardiac cells one day, but way before they become cardiac cells. And we, engineer initially some genetic construct, and after that we find cell surface marker to isolate those cells from pluripotent cells and put them back in this petri dish. And same thing, when we isolate this progenitor, you see all these cells are beating in the petri dish. And so we really enrich, capture, if you wish, the cells that will eventually give rise to these cardiac cells. So it was very important, and we can still use these to generate cardiac cells on demand and eventually to mimic disease or potentially, I will show you that in a moment, that we can use these cells eventually to re-inject to the patient that had a cardiac disease. So I will show you that. So also what we realize that this gene that we discover has the molecular switch to make cardiac cells was super important also to migrate, to make this cardiac progenitor to migrate at the good place at the right time. Because of course during development you need to adopt an identity of the cells that you want, but you need also to be at the good place at the right time. And so that's where coupling migration and cell-fate specification, cell identity, is very important. And our gene, our favorite gene here, MESP1, is doing all this. It's controlling all aspects of cardiac progenitor specification, migration, and differentiation. So after that, we wanted, because all this was done in petri dish, and so of course it's always difficult to make a very strong conclusion about what you see in petri dish, whether this is important in reality during embryonic development. So what we did with Fabienne, Samira, and Xionghui is to try to label a single cells during embryonic development and assess the contribution of these single cells to that's a heart, that's the left ventricle, the right ventricle, and the atrium, the different cavity and region of the heart. And what we found here, you see all these green or red patches. It's the progeny of a single cells during embryonic development. And what we found is, that actually this green or red patch, they were always localized to a certain part, but we never seen the contribution of a single cells to all region of the heart, and not all cell type of the heart. So it means that at the early stage of development, somehow the cells already know what they will come and to which region of the heart and which cell type in the heart they will contribute to. In addition, we found something very surprising. We found a single cells here, that were contributing both to the formation of the heart region, but also to the head muscles. So somehow you have a common progenitor for totally different structure that's very long distance apart, showing also the plasticity and early lineage determination of that. And so with all these data, we could somehow revisit the early step of embryonic development and cardiac differentiation, which was something that we found. And you see here, it's again a different heart labeled with many different colors. And all these different colors here, all these different patches, they come from a single cell. So knowing, what is the size of all these patches, we could infer what was the number of progenitor that you need during embryonic development to make a heart. And we realized that we only need about 250 cells, cardiac progenitor at single time of embryonic development to make all the structure of the heart. And so then we went to go further and try to understand at the earliest step of development, what would, since we knew that they would only contribute to one of the other region or one or the other cell type in the heart, how this molecular specification occur. And so for that we use a new technique that you're going to hear more and more in the coming years, which has been named by Science magazine as the breakthrough of the 2018 breakthrough of the year. It's to interrogate for each single cell all the genes that they express at a given time point. And so it means that we are composed of about 30,000 genes, and so each cell has expressed a fraction of those genes, and it's only the combination of all these genes that makes a cell unique. And the single cells allow you to interrogate every single cells on their own and try to find what they are similar and what they are different in between them. And so what we found here in this little red, green, and black and blue, it was already the destination where these cells, at the early step of development, will eventually go, find the destination, their future destiny, if you wish, during development. And so what I show you here is, in this diagram that, represented like a hill and a valley, and the valley is, more you roll down, so when you are at the top of the hill, everything is possible. When you roll down to the hill, you are more and more constrained in what you become and to adopt a particular identity. And what we found, and we dissect all these different steps that constrain the cardiac development at the early stage of development. And so, of course, what I told you is these embryonic stem cells, when you promote their differentiation into cardiac cells, you can potentially repair cardiac disease. And so here it's an experiment that they did on primate, because you need unfortunately, some model like that to show the proof of principle before moving to the human. And all these green cells, that's a section of the heart of primate that had been injected with stem cell in their heart, and you see all these green cells has been integrated into these cardiac muscles, showing the feasibility and the long-term engraftment of these stem cells derived cardiac cells, showing that is possible, and probably that will be one day available in clinics. Now I will discuss with you, the adult stem cells. As I said, most adult tissue constantly renew throughout their life, and they present also the ability to repair the tissue after injury. You know that if you cut yourself, you're going to very rapidly heal and repair the wound that you created. And so my laboratory is also very interested in understanding how you maintain the different cell types that constitute our adult tissue, how these cell types are, recruited and activated during tissue repair, and how they regenerate the different tissue following injury. And what we are, what has been used in the last decades as a tool to really study those questions was what we call lineage tracing. So lineage tracing is a very kind of simple, concept in which you label one cells and irreversibly with something that you can track. And here, for example, it's fluorescent protein, and I will show you a lot of fluorescent image. And so then all the progeny of a cell that has been marked is express a single color, and so you can infer a lot of things from that. And so what we realize, and the skin epidermis is a tissue like this, where you have the skin barrier, the one that isolates you from the outside, and so that's a hair follicles. And the hair follicles is, as you know, regenerating hair. And I'm sure you have noticed that when you take a shower or bath, you find some hair in your bath. But on the other hand, you don't get bald every day more and more. So it means that for one hair that falls, one hair regrows. And so indeed, the stem cells that are present in the hair follicle, they regenerate hair to make them regrow in an everyday basis. And so we knew that the hair follicle stem cell reside in this region. And what we show when I was, postdoc in New York and in the United States, is that the stem cell of the hair follicle, they rapidly migrate out of the thing to repair the wound. And what I show in my early days of skin biologist, skin stem cell biologist, is that the single stem cells can reform all the cell type of the skin and reform a tuft of hair. And so when we published that, it was kind of a really breakthrough, and everybody was very, in particular all the bald people were very, super excited about that, and we received hundreds of emails from everybody in the world. Can you, graft us some cells and remake a tuft of hair like that in my poor bald skull? And we say, no, it's a little bit too early, but indeed, it's probably more and more possible those days to do this kind of thing. So another thing that, that's very important, and is very well conserved for all evolution, because it's probably something that we've been, struggling with for millions of years, is to rapidly wound our skin, because, all the animals, they fight with each other, they bite, and when they bite, if you don't repair that, you get infected, and if you get infected, you're going to die, because, in the early days, there was no antibiotics. So it was very important to rapidly repair the wounded skin. And there's, different repair, etc. And what my lab has shown, again, with using lineage tracing, is the stem cells can migrate from very long distance. You see here is a single stem cell that has migrated from centimeters away to the wound region and repair that, and had contributed to the long-term of wound repair following wounding. And what we show, and other people show, is actually in the skin when you induce wound, actually all stem cells, and there's many different types of stem cells, they all recruit and orchestrate to repair the wound as fast as possible. And so there's a big plasticity. Somehow the cells that normally do have follicle, they don't care. They migrate and repair the skin of the epidermis. And same thing for the cells that make the greasy aspect of the hair, the sebaceous gland, the stem cell, they differentiate and repair the wound. So there's a resilience of the tissue, and the stem cells are really plastic to repair their wound. And so we, kind of trying to understand what exactly is the scaffold protein and the gene that are orchestrate this wound repair mechanism. And we are still, working actively on that. And one of the things that we are trying to understand for the moment, there's a cooking study, is when we make an excess of skin, such as, for example, upon breast reconstruction surgery following, mastectomy for breast cancer, how we generate an excess of skin. That's another kind of wound repair or tissue regeneration that we are currently studying for the moment. And of course, all this is not only to understand things. The skin stem cell has been used for decades, and it has saved thousands of lives throughout the world. And the first demonstration that you can save life was by taking that's a petri dish in which people had cultured the skin stem cell, and all these little dots here are colonies, progeny of the skin stem cell. And so following their culture in vitro, what they make is a sheet of tissue by let them differentiate it. And for a patient that has been severely burned, like 90% burning, if you don't recover the skin, you're going to die because, you cannot keep all the water in and all the microbes gets in your body. It's a nightmare. So we need to repair the skin very urgently. And so in the patient that has been burned, let's say at 90%, there's still 10% of skin that you can use. So what the doctor has done is taking this piece of skin, culture them in vitro and make this sheet of skin and reform and recover all and transplant the sheet of skin in all the burn region. And indeed, there's been many lives that have been saved through this technique. And so now what my colleague Michele De Luca in Italy has done is use the same principle, but now to treat patients with severe genetic skin disorders. So there's a genetic skin disorder that's called epidermolysis bullosa. And so those patients are extremely sensitive to any pressure. So when they put their thing like that, they make a little wound there. It's very, very painful. And there's been a beautiful story that has been published last year where there was a refugee, a Syrian refugee, stuck at the border, he lost almost all of his skin. And he was stuck in Germany. And so the German authority saw this poor kid, in a very poor shape, and he called him and said, well, Michele, can you come here in Germany with your team from Italy and help to treat these kids? And so they come, and it remained almost no skin to these young kids at all. And so he took the few skin that he could, make this sheet of cells after having correct the gene abnormality that he was suffering, and make all this sheet of skin with the genetic corrected information and graft that back to this kid. And so this kid, although, all the doctors were, were almost thinking that he was dying, he left the hospital and is now back in school and playing football with all these kids, showing that, all these things, the research I'm telling you here, sometimes it can save lives and could be very important and very precious. And so that, it's another thing that you can do with this skin, that's not skin stem cells, that's, corneal stem cells. As you know, your eyes is covered by a cornea. And that's a poor patient that has been put some chemicals in his eye and completely burned his eye, and he could not see anything. And so what the doctor has done here, they remove all this bad part of the cornea, took his stem cell from the eye, and reformed a little sheet of cell that I show you for the skin and graft that on the cornea. And that's the same patient, believe me, the same patient, a year later with from 0 vision to 10 of 10 vision. So really, again, showing the importance of the therapeutical application. So now, that's all the good side of the stem cell. The stem cell can be, important to regenerate your tissue. They can be important to treat disease, but also sometimes there's a dark side. And the dark side, one of the dark side of the stem cell is they can be at the origin of cancer, because they live much longer than the other cell, and there is more time to accumulate, the bad mutation that cells require to induce cancer formation. And my lab has done, studied the last 10 to 12 years already, what are the cells at the origin of cancer? And that was, very not known at all 12 years ago, and I think we've made a lot of progress with that. And Khalil and Gal were among the first researchers in my lab trying to address this question. And the way we did that was very simple as well, the concept. We put the bad gene that induces cancer in different cell types of the body and try simply to see whether some cells are more competent than others to induce cancer formation. And when we found out, we're trying to understand why, these cells are more competent. And then by understanding why some cells, the cell A is not competent and why the cell C is competent, then you can have some idea how to prevent cancer formation. And I will give you one or two examples for that. And at the same time, you know, we were looking at how the mammary gland, the breast tissue is developing, and we really, you know, found out that there was two different stem cells that were at the top of the hierarchy of the different cells that are present in the breast. But the breast cancer is an heterogeneous type of disease, actually. It's very frequent cancer. It will affect, you know, 1 in 8 women, so it's very, very frequent. And there's not a single disease. Breast cancer, you know, is very heterogeneous disease. And some breast cancer are of good prognosis, some breast cancer are of poor prognosis. What dictates this cell outcome? And what we did with Alexandra and Mehin in the lab is to put the bad gene either in this cell type, the luminal cells, or in this cell type, the basal cells. And what we realized, in contrast to what we were expecting, because there's, let's say, the good breast cancer are called luminal cancer, and the bad breast cancer are called the basal breast cancer. And somehow, it was totally the reverse that we found. The bad gene, when you put them in the basal cells, will give rise to the good cancer. And the bad gene, when you put them in the luminal cells, will give rise to the basal cancer, the bad cancer. And so somehow showing that, you know, the cell origin of a cancer is very important to dictate the tumor outcome and what's going to happen in the patient. And in addition, what the cancer look like cannot be used to predict what are their cell of origin. Very often you're wrong, and you know, we have many examples in the lab where everybody was expecting that this is the cell of origin, and it was not at all this one that we eventually found. And so we also look at that in the most frequent cancer in human, which is the basal cell carcinoma, which affect, you know, 5,000 of patients per year throughout the world. And you know, it looks like this little lesion, and this lesion can evolve and progress. And the question was, this lesion also, they look like a, you know, like a messed up hair follicle. So people were thinking that they were hair follicle derived, but actually not at all. We found that actually they originate from the normal cells that make the skin barrier, and they are reprogrammed into these cells that are forming this invasive tumor. And a very important concept that we learn along the way, it's actually the bad gene that causes the cancer. Somehow, again, do the reversion in time of the adult cells and reprogram these cells into a little bit their embryonic counterpart before the cells can progress to make the cancer. So again, showing you that if you understand precisely the mechanism that is behind at the early step of tumorigenesis, you may, you know, prevent tumor initiation and potentially come with very good preventive strategy. That is not necessarily, will not necessarily help to cure cancer, although, you know, sometimes there's similarity, but certainly to prevent cancer formation. So I show you, you know, that the repair of the skin are mediated by this, you know, long-term stem cells here in red, and the short that I didn't show you, the short-lived cells, the one that are replenish in constantly the skin epidermis during the normal circumstance, they are not good to repair the tissue. And what Adriana wants to know is the progenitor of the stem cells that are at the origin of this skin cancer, the basal cell carcinoma. And what she found is she found that only the stem cell and not the progenitor. So sometimes even within a tissue, two cells that almost look identical a priori can be very different in their ability to induce cancer formation. And she found the mechanism by which the progenitor cannot progress to cancer formation. Again, giving hope that if we target the genes that are present here in these red cells and not here in these blue cells, we can also prevent tumor formation. So now I will discuss with you, you know, once cancer arises, not all cells look similar when you look them at the microscope. That is called tumor heterogeneity. the tumor heterogeneity, and I will show you some example, can be very important for tumor growth, but also for the ability of the tumor to leave the primary tumor and give rise to what is called metastasis, and also potentially is important to in the therapy, because not all cells respond equally to chemo or radiotherapy. And so understanding why some cells respond well to chemotherapy and not well to the chemotherapy can be very important to improve our treatment of patients with cancer. And this is the concept of the cancer stem cells. So the fact that only few cells within the tumor are sitting also on the top of the hierarchy and do all this job that the normal cells do. And what we had an idea some years ago to try to visualize that in a real time, because that was, kind of a little bit of theoretical notion at that time, that no one has visualized stem cell inside you in action. And what we did is, so you have to imagine that if your tumor is small like that and will grow big like that, and you can label all tumor cells with a different color. If all cells are equipotent, so equal, you will see that their daughter cells will give rise to a fraction of the tumor that are all equal. If only some cells within the tumor has the capacity to sustain the growth of the tumor, what you will end up is seeing almost no color or only few color when the tumor had grown, but with having the dominant color present in your tumor. And so that would be supportive for that not all cells are equal and only some cell, the cancer stem cell, will present the extensive capacity to proliferate and to make the tumor grow. And that's what Gregory Dreesen has done in my lab. He labeled tumor cells, all equivalent. And what he found that most of the tumor cells that he labeled initially, they disappear over time in cancer. So this notion that all cancer cells are immortal, it's not true, Even some cancer cells will eventually disappear naturally. But what he found, the remarkable thing that he found that a single cell can give rise to this huge part of the tumor, and showing that, that's seeing for the first time inside you a cancer stem cell in action. And that was, a very important breakthrough at that time. And now in the field of cancer stem cell, everybody is using the technique that Gregory had developed at that time. And Benjamin had tried to understand what are the mechanisms that regulate the cancer stem cell in the tissue. And among the things that regulate the cancer stem cell is what is called a vascular niche. The vascular niche is the formation of new blood vessels that will provide to the cancer stem cells all the goodies, the nutriment, etc., to make the cancer cells happy and regulate their growth. And he could show that if we target the mechanism that make this niche or make this new blood vessel, we can shrink the tumor and have a very important impact to the tumor growth. Another thing that we were very interested to understand is to see what are the cells that give rise to metastasis. Because as you may know, this cancer rarely is the primary cancer, where the cancer appear that will kill eventually the patient. What kills the patient is usually the cells that the cancer sends in distant organ, which is called metastasis, and that is harmful for the patient. And so what we track here is this step where the tumor cells detach from each other, migrate out of the tumor and reach the blood vessels. And we try to understand whether this step of leaving out the tumor and go to the blood stream to reach their distant organ, go through different steps. And what Eugenia in the lab found, she found that there's 6 stages of the tumor's progression in which you can see their progression toward the fact to leave the tissue. And what she found remarkably is there's only 2 cell populations among these many different cell populations that give rise to the metastasis. And it's the early step of transition. And so that gives you some cue to which are the cells that we need to target to prevent metastasis. And we already found one molecule that can block this tumor transition stage and remarkably inhibit metastasis. And so we think that this new molecule that we uncover here could be used in the future to prevent metastasis in patients at high risk of doing metastasis. So what I show you here is not all cells are equal. Some cells are dividing rapidly. Some cells give rise to the metastasis. Some cells are important to invade the tissue. And so it's understanding this heterogeneity that will make some progress in cancer therapy. And so finally, what Soufiane in my lab has done is to show the proof of principle that if we target just a small fraction of the tumor cells, we can make the tumor disappear. And so here it was a technique that allows us just to kill a small fraction. And as you see here, the same animals with tumors, and 1 week later, when we killed that, most of the tumor had completely disappeared and had, and will never reoccur. And showing the proof of principle that by targeting only a small fraction of the cells, we can have a huge impact on the tumor course. And so finally, I will show you, our kind of last discovery, in which was to try to understand what are the mechanisms that mediate the resistance to therapy. And so that's, a skin cancer, that one is very advanced. Only a fraction of the patient will completely show their tumor disappear, but most of them will have some remnant cells and eventually relapse after the therapy. And so we found that in this remnant of the tumor, the one that resists to the therapy, they activate a particular signaling pathway. And so when we decide to target the normal drugs plus a new drugs that target the signaling pathway that we found in this resisting lesion, we could completely eradicate the tumor in 95% of the case. And we think that since the new combination of the treatment that we discover is immediately available, we think that, hopefully the pharmaceutical industry can launch clinical trials already today, if they wish. So, no, I give you a lot of information. I thank all the people that had done the work that I present to you. Also, I thank all the funders that give us some money to support our research. So, to do all this type of research, it needs a lot of technical support. It needs a lot of funders that come from all kind of possibility, the Belgium, the European funders, the private funders, which is you, Fondation pour le Cancer and Télévis, is you, through your donation, help us to make the work that we do to try to advance and make the progression of the science. And of course, I encourage you to keep donate for those foundations such as Fondation pour le Cancer or Télévis. And with that, I thank you for your attention. I will be happy to answer your questions.